So
That's
yeah, we are live on YouTube now, and let us start. Let me welcome Dr. Jyotsna Rao and Dr. Sarita to introduce Dr. Jyotsna Rao to the students and also to the public.
Hello, Dr. Jyotsna. It is my privilege to welcome Dr. Jyotsna, who is Senior Principal Investigator at Biocon Bristol Myers Squibb Research Center. She completed her PhD from Indian Institute of Science. She also had collaboration with Radboud University Medical Center, Netherlands. So she has been she had been a PhD researcher in both the institutes, IAC and Radboud University Medical Center. Post her PhD, she has worked as a postdoctoral associate as well as a consultant scientist in Yale School of Medicine, USA. So she is currently working as a research associate, Cancer Research UK Cambridge Institute, University of Cambridge, UK. She has received several fundings, also several awards to her credit. Few of them she has obtained the visiting fellowship through MIT DBT India Visiting Scholarship Program. She had done it along with Manipal University. She has obtained funding through biomarker challenge run by Open Plant in 2017. So she has several awards to her. Nationally accredited peer-reviewed journal.
Maybe there is a break, Dr. Sarita. There's a break in your voice.
Okay. Am I audible now? Clear? Better?
Yeah, now better. Now better.
So she has, should I, can I repeat from the funding part?
You have to report from the funding part.
Yeah. She has received several funds for her research activities, and she has several awards to her credit. She has obtained Outstanding Poster Award. She has obtained the Visiting Fellowship Award through MIT and DBT, the India Visiting Scholarship Program. She has obtained funding through Biomarker Challenge run by Open Plant to apply for ion sensing to detect apoptosis. So she has several awards to her credit. In addition to that, she has published quite a lot of articles in several peer-reviewed journals, both national and international. She has around 16 publications to her credit. She has been an oral presenter in several conferences. It's our privilege to have you as a speaker today in the session themed as in our session today. So thank you, Dr. Jyotsna. I welcome you on board.
We welcome you for your presentation on new methodologies for drug discovery, which is more awaited by the students and researchers. We want to know what are the approaches, the new approaches we can do for drug discovery. Welcome, Jyotsna, and the mic is over to you.
Thank you so much. Am I audible?
You're audible. You're audible.
Yeah. Thanks, Charita, for your kind introduction. There are a few things that I would like to say at the outset. So first thing is that I request all the participants to kindly keep themselves on mute. Otherwise, if there is any echo or something, it might disturb other listeners. So this is my humble request. The other things that I would like to say is that I would like to keep this webinar very interactive. So at specific, places, I will give breaks. So let me put the, slides on the slideshow mode first. So, okay. So, at those junctures, please feel free to ask your questions. So, or what we can do is, request the participants to put their questions in the chat box, um, so that one of you, either Vidya or Charita or somebody can actually, collect those questions and, ask those questions. So I want to keep this session very interactive, as interactive as possible for a webinar. So let me begin. So today I'll be talking about new approaches in early drug discovery. So basically, the first thing that I would like to say is that all the information in today's in this internet era is out there in the public domain. Most of the information. So actually, what is it that a webinar or somebody coming from a specific domain can tell you? It is actually what they can tell you is the way they approach a specific problem, or the way they think, or they go about doing a specific task. So that kind of information is something that is, I feel, unique and should be what is to be shared in a academic or a professional context. So in this webinar, what I'm going to do is exactly that. So I'm going to be discussing new approaches. So what are the latest approaches in the field of drug discovery, especially early drug discovery. So I'll be focusing on a very small part of the whole drug discovery process and cycle. Okay. So yeah. So this is the plan that I have for the webinar. So first I will start with the definition of a drug, and then I'll go into some history to give you a perspective of where we were initially, in the field of drug discovery, and then where we have reached now, and how our approach has changed. And that is very important, in terms of understanding, and in terms of even, applying our knowledge, and even to understand what is going on outside in the real world. So then I talk about the process of drug development. What are the specific steps? You know, and then I'll talk about specific things like target identification, selection, and validation. Followed by lead discovery and optimization. These are the crucial things that I would like you to take away from this webinar. And then I'll summarize the whole talk. And at the end, I will give the hackathon problem as I was requested. So now let me tell one more thing, that this is a very vast topic to cover. And I'm keeping this at an introductory level, and I'll go over the basics in detail and spend a lot of time on each of the aspects. So at the end of this webinar, the key takeaway for you will be an understanding of the basic processes involved in the drug discovery, early drug discovery, and what are the new technologies that one can apply. So what those specific technologies are, there's enough information available on the public domain. So, details of those technologies, because, otherwise it becomes way too complex to go over multiple technologies. So now let me begin by giving you a definition of a drug. What is a drug? So this is something that you people might be actually hearing very often these days in news. So a drug is a chemical that brings about a change in biological function through its chemical actions. That is the most simple definition of drug. And this definition of drug, has kind of this can be applied both for pharmacological drugs and psychedelic drugs. So it just brings about a change in biological function. So how did people start thinking about the whole process of drug discovery? And that is something very interesting. So this has what you may call as ethnobotanical origins. What do I mean by ethnobotanical origins? Something like our Ayurveda, Siddha, or even homeopathy. So all these are traditional medicines, and people used to use plant extracts and realized that a lot of these plant extracts had medicinal properties, and they could be used to achieve a specific, change in the biological function. For example, pain relief. It's a very very simple thing for which a lot of traditional medicines are available. So that is what I mean by ethnobotanical origin. So extracts of barks, extracts of nuts, extracts of different plant parts were being used for a long time in human history. But all these approaches are what you can call as mechanism agnostic approaches. So people did not exactly know how these plant parts acted. Okay, so these plant extracts acted. So they were mechanism agnostic. That's what they call it as. But people were really interested in trying to find out how they acted. So they started, kind of simplifying the extract and trying to, home in on a specific compound that could be present in these extracts, which could be the effective compound and which could be the one that is acting to produce a desired change in the biological function. So that actually gave rise to what is called as chemocentric approach. The chemocentric approach is how a specific compound or a class of compounds could influence a biological function to see a desired end effect. Okay, so then they started further optimizing these compounds. So, for example, there are many stories that I can tell you here. One famous story is that of aspirin. So, aspirin is was actually initially developed from the extract of the bark of willow tree. Willow tree is Salix alba, and that is how the name salicin. So there was one compound in the extract of the bark of willow tree called salicin, and this salicin, could actually have pain relieving effects. So people were using the bark of willow tree, the extracts of the bark of willow tree, to actually treat, to cause pain, to get pain relief. So these, as they started distilling down to this compound called salicin, they realized that this is actually the effective molecule that is bringing about the effect. Now, uh, it was not exactly tolerated by everybody. Okay, so in its original form, salicin was not tolerated by everybody. So there was a scientist called Felix Hoffmann in Germany. His father had a rheumatoid arthritis, and he used to be in a lot of pain. So, and he did not tolerate salicin. So now, what Felix Hoffmann did was to try and improve the formulation of salicin, trying to get it better and better. So he synthesized salicylic acid and then acetyl salicylic acid. So this acetyl salicylic acid, which was synthesized by Felix Hoffmann in 1899, is what we now consume as aspirin. But people did not know how actually aspirin acted. So there was a 75 years wait time before the link between chemical derivatization and the therapeutic effect of aspirin could be understood. So basically, um, this basically how did it work out then? How, without even knowing that this derivatization of as salicin into acetyl salicylic acid could be the one that could have the therapeutic effect. It was just serendipity. And in most of the things, the ethnobotanical origin compounds, which later found their mechanism of actions or their derivatizations on mechanisms of action. They were all successful even before people knew how they acted. So serendipity had a huge role to play there. They were, I mean, simple words, they were playing lucky. So this continued for a long time until the advent of biochemistry and molecular biology, and until the time, and after the advent from 1945 50s, with the development of the field of biochemistry and molecular biology, people started applying those, the learnings into the field of pharmacology. So, in mid 80s, molecular basis of pharmacology started gaining a lot of ground, and that is how now we actually go about the whole process in a very informed way, and that is, what I would like to present today. So this is the perspective. Now, were there other drugs, or medicines which came about in the same way? Yes, there are many. One famous story is that of penicillin, which you all might know, and also, for example, the isolation of steroid hormones from urine of pregnant women, which actually led to the development of contraceptives. So these are all, very famous examples of how, uh, people started developing drugs without the complete knowledge of molecular basis of their actions. So now let me move on to the process of drug development itself. So when you start out with the process of drug development, there's the potential new drugs, the number of potential new drugs is astronomical, literally astronomical. So it's a huge number of compounds, or a huge chemical space and a huge target space that you are actually sampling. So the whole process of drug discovery thus becomes a very long drawn process. So you can see here that it might take anywhere up to 15 to 20 years for getting an approved drug into the market. All right. So it's a very long drawn process, and the success rate is very less. So it's under, 5% that there is a success. Okay. And here, most of another thing that I forgot to say is that most of my talk is going to be focused on small molecules, because that is the field I am in. It is heavily influenced by that. But the same thing applies for biologics, as well as, for example, vaccines and other things which you might be very much interested in due to the given COVID situation. But it is the same kind of pipeline, it is the same kind of timelines that apply everywhere for all kinds of drug development. So there are different steps in the process of drug development that have been identified. There is nothing like, there are no silos here, but these are like rough compartments, if I can use the colloquial language. So it's the borders are rather dim. So there is the phase of early drug discovery, which involves identification of a target. So what is it that we can target in the cellular physiology that can produce the desired effect? Okay, so there is the target identification and validating that target, and then discovering the molecule that actually can hit the target, or a set of molecules rather, that can hit the target, and develop assays and screening methodologies which can actually verify that. For example, high throughput screening. And then we actually then go over to what is called as hit to lead, which means that what are these uh, specific set of targets, hits, which can be uh, you know targeted by a set of compounds. All right. So, for example, if there is a signaling pathway in the cell, what is the set of targets that you can go after? What is the set of lead molecules that can actually engage those targets? and then optimize the lead. So this entire whole process is what is actually the process of early drug discovery, and this will be the focus of my talk. All right. So it's just a very small and the very early steps of drug discovery. Now, once the lead compounds, set of lead compounds are optimized, then they are taken into in vivo, in vitro, and ex vivo assays. That is, they are tried in animals. They are again tried in different sets of cell lines with a more, you know, preclinical kind of a focus, not development focus. Then you're trying to look at specific ex vivo assays. That is, you can take out an organ and see what is the effect. Then you're looking at the absorption, digestion, metabolism, and excretion. And in fact, I should even add T here, that is toxicology. ADMET is how pharmacologists call it. Okay, so people look at that for the different set of compounds. Okay, and then they do proof of concept studies and think about how this drug can be delivered. What is the safest way to deliver the drug? How it should be formulated, how the formulation should be optimized um, in a way that it achieves the maximum bioavailability with minimum toxic effects. Okay, that is side effects. Okay, then you find the range of doses, and then you file an IND application. Say that this is a candidate. Okay, now you start the clinical development where you try it on either a small set of patient population or a healthy volunteer study to see the toxicological effects of the drug, and then in the phase 2 and 3 it is studied, the drug effects are studied on different sizes, increasing sizes of patient population, and then people see the dose effect, uh, uh, you know single dose, ascending dose, multiple dose studies, safety, efficacy of the drug, what is the pharmacokinetics of the drug, you know, a lot of all these steps, they come under clinical development. Actually, I have very limited exposure to clinical development of the drug. That's probably something that, people who are more into clinical trials can help with. Then, it is filed for approval. Then the whole process of review happens. For example, in US, the FDA review happens, and then there are the drug either gets approved or fails to get approval. Whatever the decision is taken. So to get to the approved drug, you actually go through this entire process. So it's already like 10-12 years into the whole process by the time you have an approved drug. All right. So am I still audible, and does anyone have any questions at this point of time?
You are audible, ma'am, loud and clear.
Okay. So is there any question that anybody has? Okay, I'll continue then. All right. So now we go on. So in this early drug discovery process, so I have shown you. Okay, I've already taken quite a bit of time. So is it all right if I go a bit over 11? Is that all right?
Sure, ma'am, you can continue.
Okay, all right. So now in the as I have described here in the early drug discovery, the first step is target identification and validation. So one famous saying by the father of modern pharmacology, Paul Ehrlich, is that drugs will not act unless they are bound. It's as simple as that. So basically, for a drug to act, it has to be bound to a target. Now the question is that how do we find the target to which you will design a drug molecule, or find a target molecule for an existing drug. All right. So the drug target is amenable. So what are the features of a drug target? It should be amenable to modulation, and it should be relevant to the disease phenotype. What I mean by that is that we should be able to modulate the target with the specific compound and with a specific compound, and then that modulation of that protein, for example, should be relevant to the disease phenotype, and we should see the biological effect. So this actually thus involves deep understanding of the clinical spectrum of the disease and a deep understanding of the molecular basis of disease pathophysiology. So how does the disease originate? People should have a very deep understanding of that. And a lot of this happens in the academic research. And people, thus the entire drug development process and the pharmaceutical companies that depend a lot on the academic research that happens. Okay, so the process of target identification thus involves extensive review of literature. So even in my workplace where we propose targets, actually we do a very extensive review of literature, and it's a whole bunch of papers which we look at it in a very objective fashion. So how well we can use a specific protein, for example, as a target. So how do you consider a target as druggable? Then we should have some rules, right, for us to say that, okay, now this is how I say that this target is druggable. So that involves like 5 different key features. So first thing is that this target should have a confirmed role in disease pathophysiology. So people should know like where exactly it fits in the pathophysiology, and the expression of this target should be highest in the disease tissue than in the other tissue. So even if you provide it systemically, it should have minimum toxic or side effects. All right. Then it should have a favorable toxicity profile, that is minimum amount of off-target effects and side effects. Then it should be assayable, and it's something that the target we should be able to assay the effects. Also, in fact, it would be really nice if it will actually in today's world move forward only if it is amenable to high throughput screening assays. And then we should ideally have the availability of the 3D structure of the protein, for example, which we are targeting. So you can consider a target as druggable only if we have all these features covered. So now the process of target identification. So how do we start? We start with as I said, deep dive into relevant literature and look at public databases, like Drug Bank or TTD, where you actually get an idea of what are the existing compounds for a set of targets if you are trying to look at addressing a specific problem or a specific disease, for example. For example, a pharmaceutical company will have a specific therapeutic areas they'll be interested in. So in that therapeutic area, there will be specific diseases which they have a preference for. For example, let's say that. Then you go back and do a deep dive into relevant literature and public databases and try and figure out a match of targets and maybe possible compounds. So then this process, it can happen in 2 ways. So here you either already have a compound and now you are seeing an effect of that compound for some reason. All right, you already have a compound with you. Then you deconvolve the target and understand where exactly this compound binds so that it is having its biological effect. And then how can we improve its biological effect? Can we improve its biological effect? For example, drug. So, for example, if there is a drug that works on a specific disease, for example, cancers. It works in a specific cancer, but a similar pathway is also functional in another type of cancer. Can we deconvolve the target can we understand how exactly it is, how exactly it might work in the other type of cancer, and how we can actually improve this compound, if is it possible to do that, and get finally one target and validate that target. All right. Otherwise, there's another way to do it, that is target discovery. So this target discovery process has this philosophy that to get a new drug, you should have a new target. So you want a compound basically. Then you should know what the target is and its exact role in the disease process. So you already know about that. So you know that the specific protein, if it is modulated in this disease, might have a good effect. Then you go about synthesizing or sampling the chemical space for that compound. That means that you try out thousands or you know uh, uh, hundreds of thousands of different compounds and see how it can, whether it can bind or not. So this target identification facilitates uh, uh, the process of drug discovery, and it enables rational design of new molecules with improved safety and potency profile. So basically, this whole process now looks far more rational than what happened in the story of aspirin, right? So this is the way people approach target identification now. Okay, now let me go on to the first process of deconvolution, target deconvolution. So this process of target deconvolution uses phenotypic approach to drug discovery. So what do I mean by phenotypic approach? It actually that whatever compound should have a effect on the phenotype. So you should see a change in the cellular phenotype as a result of adding that specific compound, and it does not rely on the identity of the specific drug target. Basically, you want to see the effect. You don't, at the first time, you don't want to know where exactly it is binding. You want to know first whether you are seeing the desired effect. For example, cytotoxicity. Again, coming from the cancer perspective. Okay, you want to see that it is killing the cells. Basically, that's it. So for this, a set of phenotypic assays is performed, and this phenotypic assays will identify a compound's ability to alter a cell's phenotype. For example, growth arrest. For example, death of cells. So these phenotypic assays can be performed in vivo or in vitro. So in vivo is that you have a lot of compounds and then you're testing with them on preclinical models of disease. But this is very low throughput and ethically unjustifiable. How can we justify trying out, even at the very beginning stage, hundreds of thousands of compounds on animals? That's simply ethically unjustifiable. So people use what is called as a cell-based assay approach to provide the cellular context. So this cell-based assay approach provides the cellular context and it gives the physiologic relevance, and it is also compatible with high throughput screening. What do I mean by high throughput? So you people might be aware of 96 well plates. So each of these wells here serves as a small tube, all right? It's like a small test tube. So basically, that then says that the assay should become smaller and smaller, and it should be done in smaller and smaller volume. So miniaturization comes into picture. So this is a 96 well plate, and next you see a 384 well plate, and then you see a 1536. 1536 well plate. So these plates help in doing the assay in a high throughput format, and they help in miniaturizing the assay. Okay, so thus by doing this target deconvolution using a cell-based approach, cell-based assay approach is uh, makes it very effective in terms of screening lots of compounds. And now they also we are trying to do new things so that they also reflect the in vivo environment. For example, 3D cell culture. So what is 3D cell culture? The cells are now no longer attached to plastic, which is a very artificial way of growing cells. They are grown as spheroids. as a ball of cells. So these spheroids are derived from patients, or they are derived from cell lines, established cell lines, and then they are grown in these different high throughput plates, and then they are screened with different kinds of drugs, or if we are looking at a specific target, or if we are trying to identify a target, then we are actually using tool compounds. So this is the other concept that I want you to know about. So you already don't have the drug now, but what you have is a tool compound which you can actually screen with. I'll come to this later. So there are other approaches called there are many other approaches that you can use, like apart from the phenotypic screening approach. You can use biochemical approaches like affinity chromatography, where a small molecule is immobilized onto the affinity column via a linker. So these things you might have come across. So when this happens, you are screening immobilized target, and then you're so you are putting the target analyte to the column, and then you are, for example, having an immobilized receptor and trying to see which one binds best. So the other techniques that people can use is phage display, mRNA display, pre-hybrid system, etc. So for example, you have a display. Phage display colony. So you can have bacteriophages with your gene of interest inserted in them, and then there are a lot of proteins displayed. You can have specific proteins displayed on specific bacteriophages, which can actually go and bind the immobilized antigen to the immobilized protein target that or immobilized antigen which this protein can go and bind to, and then you wash it. You know which exact bacteriophage is remaining, and then you reinfect the bacteria with that bacteriophage and amplify, and you go over the whole cycle again. So this is phage display. You might be aware of this. Then there is mRNA display, which involves, it is a display technique used for in vitro proteins, and you actually have a mRNA of the same protein, and there is a puromycin linkage, and then you actually synthesize that protein based on the mRNA tag. So you have the mRNA tag for the protein, and then you have the actual protein itself. So and then you are screening which which of these proteins. So you actually now have a whole set of them. So there is one compound of, for example, one kind of drug target, drug that you have. This is a deconvolution approach. So you already know the compound. So so there is a set of compounds that you already are interested in. Now you have a whole set of proteins which are bound to their own mRNAs. You go ahead and use affinity chromatography to try and screen which of these compounds were bound by those specific proteins. So which proteins finally bound to that compound, you can actually decide that. And then because you have the mRNA tag, you already know now which is the actual protein that was eliciting the effect. So this is how you can go backward and find out the target. So that is the deconvolution approach. So there are other methods that are used. For example, protein microarray. So with the advent of the robotic printing on membranes or glass slides, the whole field of microarrays evolved. So basically what people do here is that they immobilize a native protein or an antibody or a peptide on a glass slide or a nitrocellulose membrane or any other kind of synthetic membrane, and then they block all the non-reactive sites, and then the sample is incubated, which gives the specific binding. So now, for example, your drug compound, whatever is of your interest, so where will it go and bind? without labeling, you can detect. So if this compound is not labeled with any other fluorophore, it can be detected with mass spec or surface plasmon resonance or atomic force microscopy. I mean, these are not high throughput techniques. SPR is, mass spec is, but not AFM. So MS and SPR, you can use that, or you can use fluorescence-based detection, which is actually far more practical in terms of an industry setting. And then you're detecting the specific labeling. The other thing that you can do is something very smart that was done by Peterson et al. In 2006. There's a very nice paper where people use a very smart approach called as biochemical suppression, where you actually have an uninhibited cytoplasmic extract. This is a cell-free extract now. Okay, and this is again my protein microarray is cell-free. So you don't have the cellular context. That is something that you should remember. But nevertheless, so you have an uninhibited cytoplasmic extract, and then you add the small molecule, and you are seeing the effect. Basically, inhibited extract. And then from the uninhibited extract, you start adding different fractions to see which fraction will actually give you back the activity. All right. So this was done on a TAR actin polymerization. So an inhibitor of actin polymerization. The target of that was found using this method. So then you add, so you understand that for example, in this picture, you have third fraction, which actually gives you the which brings the whole thing back into takes it out of inhibition. So basically now the inhibition is lost when you add the third fraction. So what does that third fraction contain? You again go about doing the whole process. So you again fractionate this third fraction and then see which of this. So finally you arrive at one molecule. So then there is another interesting thing called reverse transfected cell microarray. So what is reverse transfected cell microarray? It is a plasmid. So why do we call it reverse transfected? So again, this uses the microarray robotic DNA printing technology. But here, you have a plasmid with the promoter. With the GFP under a promoter. All right, this promoter is sensitive to the modulation of a specific signaling pathway. So there is a signaling pathway modulation of which affects this promoter, which actually can regulate the transcription of how. Okay, so I had a question there pop up. I'll finish this and I'll ask. I'll try and answer the question. So the first thing is that you actually have this plasmid DNA printed on the glass slide. This has the GFP under a promoter that is under the control of a specific signaling pathway. So now it also has the cDNA of the protein of your interest. So now the plasmid is seeded. All right, it's unlike transfection where you first seed the cells and then transfect. First DNA is seeded on a glass slide, for example, and then the whole glass slide is incubated in the transfection agent, and then you take that off and you seed the cells, and now you see which of the parts that got transfected actually have the effect on the drug. So now you have this microarray with small dots of cells which are transfected with this like this small dots of cells which are transfected with a specific plasmid. Now you add your target drug and see that you modulate a specific part of a signaling pathway, whether that drug has any effect on that. So you will see that with the expression of the GFP. Okay, so your protein so you think that it might be this protein, or you think that it might be the other protein which is under that promoter, whether that or which of those proteins was actually bringing about the effect, you will know that. So this is called as reverse transfected cell microarray. So these are the different kinds of technologies which you can actually use for target deconvolution. So I'll take the question. Can somebody say the question out? It kind of disappeared from the screen here.
Ma'am,
Akshay, could you please?
would you like to answer all the three?
Yes.
Okay.
Sorry.
So the first one is how accurate and cost effective is 3D cell culture?
How operationally cost effective? They are trying to make it as, for example, we are trying to develop a 3D cell culture platform, and that we are trying to make it as operationally cost effective as possible. But it cannot be more cost effective than a biochemical approach. Any phenotypic screening is costlier than a biochemical screen. That much I can say. But nevertheless, that kind of, how do you say, your chances of success will be higher using a 3D cell culture approach than a 2D cell culture approach. Actually, I can give you examples for that. Their metabolism changes, the cells, the way they metabolize changes, you can also get a glimpse of the effect of penetration. So the cells which are growing in 2D are not exactly what you have in the biological context, right? So it has a huge set of advantages. So which is why, though it is expensive, people are trying to use it.
Okay, thank you. The second one is what is the criteria for choosing each of the three expression cloning systems?
What is the criteria for
Choosing each of the three expression cloning systems, what you mentioned.
Okay, so what is the criteria? So actually, first criteria is what is it that you can practically do in your setting. That is the first criteria. Second criteria is, for example, expression cloning system for mRNA display or phage display. It's interesting. Basically, what we are in a practical setting, what we are doing is what we can actually practically do in the lab. But if you are having, for example, bigger, so what is it that you can effectively? mRNA display system is a much more, so it uses in vitro transcription, right? So the way the proteins are expressed is does not depend on the bacterial cellular context. All right. When you have a feeling that your actual protein is not completely translated uh, in a bacterial cellular context, because their translational machinery is different. Okay, and then at that point of time you decide which one do you choose. So for example, even with mRNA display, I'll tell you something. These three hybrid system is probably much better because this is a eukaryote, all right, and you can probably get some amount of glycosylation. But with the prokaryotic system, you don't exactly express the proteins the same way. So I would say that if you want the most physiological kind of protein expressed, which can actually try and bind your target, then you would probably use a three hybrid system. But then you can many times do away without that. We can many times use just an in vitro transcription translation system to get your protein of interest, and that will still be fine, where it is not heavily dependent on, for example, glycosylation, on some post-translational modification that you cannot achieve in a tube, for example. So these are the criteria. So that is why having some knowledge of what you're looking for is very important. If you think, for example, you have some kind of an idea, like this, it could be this protein, then what is the glycosylation, or what is post-translational modification that is happening? All these things become very crucial. So, what was the next question?
Just a follow-up for that question from my end. So these PPs and all those things can't it be predicted using software?
We can. I'm coming to that.
Okay. So the third question is one, do all these steps of target deconvolution needs to be followed for repurposing of drugs? or is there a way around it?
No. You see, you apply what suits you best. So not all these approaches are applied all the time. You can choose the approach based on what you can do actually, or based on your target, based on your compound, based on the prior knowledge that you have. So actually, you don't go into any process without any information, right? You already have some amount of information on which you are building up slowly. So based on that, you decide what suits you best. Because I haven't seen biochemical suppression being applied so much in industry. Protein microarray. So it depends on the setting as well. So yeah, that's how you apply. You don't have to apply all of them in series or in tandem, or no. It just
So we can just skip the few admin tests and in
not
vitro assays.
no admits are not skipped. So this is just
Okay.
at the very beginning step. This is just the target deconvolution. You just have the compound that you think it is showing some effect. Now you're trying to find a target. You are in the early stages. You haven't gone to the preclinical stage yet. So you're just there. So even at that stage, you are trying and finding where does this compound bind. That's all you're trying to do here. So even for that, you will have to convince people that no, you know what, this compound is binding here. So for convincing, you have to do a set of experiments. What are those set of experiments? It could just be a biochemical experiment. It could just be a phenotypic experiment. But usually it is a combination of both.
Okay, ma'am. Thank you. You can continue with further.
Okay. Okay. So now the other step is, target discovery. So for example, here, you by here biological targets are known ahead of the lead discovery. So you know what the target is. You think that targeting this specific protein in this specific signaling pathway is going to give you a wonderful drug. Now, for that, you should know what is the role of that target in the disease pathophysiology, and it should be very well established. Just doing this takes a lot of experiments, and that is why the whole process is such a long run process actually. And nobody wants to commit to something unless they're very very sure, because it's a very expensive process. So this at this stage, so I was mentioning about tool compounds, right? At this stage, people exploit what are called as tool compounds. For example, you know that there is a specific kinase, and there is a specific a compound that can I don't usually give out names. That's because of my job. All right, I try to keep this as generic as possible. Please excuse me about that, unless the experiment or something is cited in the literature, right? I don't specifically go about mentioning any of the targets or compounds or anything. So I am keeping it generic. If there is a kinase and there is an inhibitor for that kinase. So a lot of them are, for example, PI3 kinase inhibitors. A lot of them are available on the in Sigma. You can buy them. Selectchem, you can buy them. So these are the tool compounds. So you want to know whether inhibiting that specific kinase will elicit the kind of effect that you want to see, desirable effect. Okay. So you can exploit the available tool compounds. That is one advantage you have if you are going fresh, trying to say that this is the target that I want to approach. Now, there could be multiple approaches that you can actually simultaneously apply. So here comes the computational modeling that you are talking about. So you can actually, you know which protein you want to target. You can crystallize it. You can do computational modeling. You can solve the solution structure. Then you can take genomics approach, biochemistry based approach. You can look at the binding kinetics of a tool compound to the target and understand whether this target is something that we want to go after at all. Okay, then with all these different sets of experiments, what you actually are doing is you are enabling the determination of structure activity relationship. Okay, so once you actually determine the relationship between the structure of the protein and the activity, and targeting which part of that protein might give you a good drug, then you are in business. So also you can use many other approaches. For example, what will be the effect of reducing the expression of a specific protein? You can take siRNA approach, you can take CRISPR-Cas approach. All these are very, popular approaches. You can get a lot of information about them on the net. You can also do RNA-seq and DNA microarray kind of experiments where you have where you are inhibiting a specific target with the specific tool compound and then seeing the effect that it has on, for example, gene expression changes, which is very important actually. So how it affects the entire cellular physiology, that cellular context is important. Then you also can actually take the systems biology approach where you have the all the transcriptome, the proteome, and all the mutations, for example, associated with a cancer. You have all the information, and now you are trying and applying it to understand what could be that specific target in a specific cell signaling pathway. If you inhibit which, you will see the desired approach. Also, people these days, because of the systems biology, what has come in is artificial intelligence or machine learning approaches, where you are actually not biased. You are using machine learning tools to actually decide on which pathways could be very effective, combining all the three information like expression profiling, your proteome, and your mutation profile. All right. So all these different approaches come together, and then they give you an idea of what the target could be. Somebody had a question. Sorry. Hello. Am I audible?
Yes, ma'am.
Okay. Can somebody say the question, please? It disappeared again. Or maybe I can see it in the chat.
No, ma'am, there are no questions as of now.
Okay.
Good
All right. Okay. Then I'll continue.
Yes, ma'am.
Okay. All right. So now this whole process of target identification. So what is target validation? So you validate the target that you have identified. So you have used all these different approaches, and now you think that this could be a target. Well, there could be one protein or a signaling pathway or multiple proteins in that signaling pathway, a kinase upstream, a kinase downstream, something like that. So this target so you have that your identification process, first thing should be reproducible. So your experiments, what you have done to reach there, should be reproducible. Okay. So the next thing is you modulate the target or the target compound microenvironment. Okay. Then what I mean by that, you do something to the target so that you make it lose its ability to bind to the compounds, or your tool compound or something. So for example, you introduce a mutation. So this is like your negative control. So this shouldn't work. So basically you introduce a mutation in the protein target, so you see the activity loss. Okay. Or you see a decrease in the activity, which is modulation of activity. Okay. Or you use siRNA based approach where you are temporarily suppressing the expression of the gene. But you should remember that downregulation of a gene expression is not the same as inhibition of a protein activity. They are very different. Okay. Also, there is another problem, whereas siRNA delivery is not always very easy. Okay. The other thing is CRISPR-Cas based gene editing technology, which is very popular these days. They enable generation of activity dead mutations. So basically, you think that this proteins, this specific activity can be a target. Now you generate activity dead proteins using CRISPR-Cas based technology, and then use that for your validation routine. And basically, that protein being mutated or losing its activity should have the effect on the phenotype, right? Then you have target ID and validation, the next step. So basically, so now you have identified the target and you have validated the target. You have the whole thing ready with you, and you have verified it, and now it is actually considered suitable for pharmaceutical development. So this whole process is actually takes a good easily 3 to 5 years in a academic setting, or at least good 2 to 3 years with multiple groups being involved in a pharmaceutical setting. So now you have now you are ready to go with the pharmaceutical development. Now what you do is extensive screening of the compound libraries to identify what is the hit. So what set of compounds actually can bind to the target is what you now start looking at. Okay, this is now this is the case that I'm talking about where you already don't have the molecule. Okay, now what is a hit? It is a compound that interacts with the target. This can also be called as lead. Okay, so these are so your these are, I mean, terms which are kind of used in slightly depending on the context, used alternatively. So so you have strategies to identify the hits. So what are these strategies? So one thing that I would like to highlight is that the chemical space that we have, that we can sample, is very huge, 10 to the power of 7 actually. So that, or even more. So a huge, not 10 to the power of 7, it was even more. It's astronomical. Actually, this is the estimated number of stars in the Milky Way somewhere here. You can see the kind of chemical space that is available for people to sample. So these many compounds are theoretically possible, all right. In this chemical space, you are trying to sample uh, one compound that, or a set of compounds that can bind to your target. So there are a lot of target libraries, target directed compound libraries that are available. So some of these libraries are commercial, some of these libraries are proprietary. Commercial is what you can buy from vendors. Uh, yes, peptides are being used as inhibitors in some cases, but their bioavailability becomes a question. Okay, then proprietary, you know, uh, compound libraries. These proprietary compound libraries are actually what we are seeing in a mm, uh, pharmaceutical setting. Okay, they, for example, PMS has a proprietary library which we don't share with anybody. And then there is public, what is available in public domain. All right. Then, so these compound libraries can be screened. Also, you can perform what is called as virtual screening. What is virtual screening? Again, this is a computation-based approach which I will go into. So you can perform high throughput screening, phenotypic screening, and fragment-based screening. Phenotypic screening is what I've already gone through. So I'll talk about high throughput and fragment-based. Virtual screening. So this uses in silico approaches. Okay, to screen large databases of a chemical library. So you know this is the target that you want to go after. Specific kinase, for example. You know the 3D structure of the kinase, then you use structure-based methods, and then you see what kind of a ligand can be docked into that protein, because you already know the structure. Now, the other thing is that you know a target, that this could be a good target based on some screening, protein screening, biochemical screening, some approach. Now, but you don't know its 3D structure. Now, for not all proteins you can crystallize them, or you can know their 3D structure. Okay, it is that itself is a very challenging process. So there, what you are doing is using ligand-based methods. So what you can do is that you are choosing bits and pieces of different chemicals to try and see whether it can bind to that specific protein. If it can bind to that specific protein, then what it does is that it can actually, it kind of starts giving you an idea of what the protein might look like. So this idea of structure that you are evolving, it's evolving in your understanding based on this different ligands that this protein can bind to. Actually, constitutes what is called as pharmacophore mapping. Also, it can give you an idea based on, so there are similarity searches. So basically this ligand that you are trying to bind, that is kind of binding to the given protein, also binds to other proteins, right? For example, if that is the case, then that can give you the similarity, that can actually kind of make you understand that this protein is similar to the other protein. It is also similar to this protein. So they all have something in common. What is that common thing? So that is how you evolve the understanding of the structure. Also, if you don't know what it is binding to or what it is not binding to, then uh, for example, you know some things that it is binding to and some things it doesn't bind to, then you can use machine learning approaches. I'm not very clear how the whole process happens because I'm not very much into this whole field of machine learning for drug discovery, but they tend to use these approaches, and these days it's becoming very popular. There are a lot of people who kind of go about using this. For example, in neuroscience, they use a lot of machine learning based approaches to generate new enzymes or different kinds of enzymes. So different kind of tool enzymes for that matter. So now, now the next thing is, so your okay. So the other thing that you can do is to screen existing compounds. Okay, or you can take a completely new de novo approach. So uh, even in silico. Okay, so basically, you know what the compounds are, so you screen based on them. But you can actually sample the chemical space and say that I want a COOH group, I want that group, I want this group, specific group. So you're actually building a new compound. Okay, so when you're looking at existing compounds and applying it to virtual screening, you're actually limiting yourself. You're actually working with a limited sampling space. But advantage is that the compound is readily available if the hit is identified. You can see that in silico this compound finds, then you have already, the compound is already ready synthesized. But in the de novo approach, you create new compounds with specific groups in them. There is no limitation on virtual chemical space. All right, you can actually create a lot of new compounds, but the question is whether you can synthesize them and whether the pharmacological development of that is viable. That becomes a question. You can take either approaches, but depending on what works best, people choose. Depending on their setting. Now, people also use what is called as high throughput screening. High throughput screening is, so this is, now we are coming back to the lab. It's not virtual anymore. It's not in silico. This is in vitro, okay, or ex vivo. So you can rapidly test millions of compounds for biological activity at cellular level, pathway level, or molecular level. Okay, so so this is the most common form of screening that is applied in pharmaceutical industries. So, you can screen anywhere from 1,000 to 10 power 6 millions of small molecules. But that requires an assay design. For example, you are setting up a biochemical assay. The assay design has to be very simple and compatible with automation. So the companies use robotic liquid handlers because now you're dealing with plates of this size, 1536, those many cells. You cannot sit and pipette into those. It's not possible. 384 well plates, you cannot find into it. It's really, painful trying to pipette into 384 well plates. So people use robotic liquid handlers, and also data processing is automated. So assays are being performed in these kinds of high throughput formats. Now, how do you do this? How do you automate this? So for this, there are different commercially available high throughput platforms, automation platforms available. For example, we have what is called Genesis. There are bench cell. There's contactless liquid handler. This is Eco. So basically, Eco-based sampling, and then all if there is one from Agilent. So there's Janus from Perkinel, all of these. So, and they are also compatible with cell-based assay because they all have HEPA filtration, all right, which is needed for your cell culture, so that your it's kept contamination free. So it's very interesting. Actually, you have a lot of videos of these in public domain. You can go and watch them. They actually have heads, pipetting heads with different, formats like 1536 head, which can pick up those many pipette tips and then pipette it into the plate. 384, 96, like that. And they're interchangeable, by the way, and the whole thing happens automatically. Then you have what is called as a plate hotel, which actually holds all plates which have the cells in them. For example, if it's a phenotypic screen, nice and warm at 37 degrees. Okay, then the plate automatically comes out, the compounds, whatever you need to pipette or whatever needs to be removed from the plates, everything is either pipetted in, removed from that. All these things, happen automatically. And that makes life far more easier for a bench scientist, because now you are focusing more on, setting up the whole process in the first stage, and then you can forget about it. You have a million compounds, you can screen, there should be no problem. Okay, so this is the part for the high throughput screen. Now there is another thing called fragment based screening. Okay, so now high throughput screening is applied where you already know the compounds, right? You have a chemical library, compound library. You are screening that compound library against a target. So now you have identified the target. You are now trying to see which compound is interacting, which sets of compounds will interact. They are all, tiny variations of the same structure. So now the other way to look at it is, there is a protein which modulating which will have the effect, but you don't have one. You are going about synthesizing a compound which can actually generate a hit, generate a lead. So you have this protein, and there are these specific pockets, a kinase, for example. You have something that fits here, something that fits here, something that fits here. These three different colors. Okay. Now, if you fit an inhibitor in which can actually occupy these three spaces, then you are inhibiting that kinase. Suppose now you actually can design a chemical by linking these three different structures. So this is what is called as fragment based screening. This actually allows you to sample a much larger chemical space when compared to a very small chemical space, relatively small chemical space that you can actually sample with your regular high throughput screening with the compound library. Okay. Now these fragments are like organic molecules which are very small in size and low in molecular weight. So what that gives you is that already you're starting off. Even if these have very low binding affinity, it's all right, because together, when they come together, their binding affinity will be higher. And then the fragment based lead discovery can actually help design multi-target drugs. So you have similar pockets in other proteins, then you can actually think of using these specific fragments. That is another advantage. Okay, so it has several advantages over traditional compound library screening. It can give you more, you are sampling a larger chemical space because these are smaller, these tend to be lower in molecular weight. They are also more hydrophilic. They are actually more bioavailable that way. And then your chance of getting a final compound that is of low molecular weight is also high. And then you're actually dealing now with at the end point, you're dealing with a structure that has very low steric hindrance, because you have designed the compound in such a way that there will be minimum steric hindrance. Okay, so these are the different approaches that you are actually getting. So this to find the lead and then optimize the structure that you actually want to go after. Even here, you don't have one single compound, but you have a set of compounds which you can go further and try. So let me give you an example. So there is a disease called cystic fibrosis, which many of you might be aware of. This disease is linked to a protein called a CFTR, a cystic fibrosis transmembrane conductance regulator. This is a ion channel. Then this is a disease that is associated with mutations in this protein. And when there is this mutation, there is this impaired ability to pump chloride ion or other ion across the cell membrane. So there are more than 1,700 mutations linked to the impairment of CFTR. Okay. Now, one pharmaceutical company, this is available in public domain, all can look it up, how Vertex Pharmaceuticals came up with Ivacaftor. So Vertex Pharmaceuticals is a company which actually came up with a drug called Ivacaftor, which actually had a very good effect on a specific set of patients with CFTR. Okay, so they came up with a orally bioavailable. So these are very important things to remember. They're orally bioavailable, so it's easy for the patients to take the drug. So orally available small molecule, and that halted the disease progression. There's no solution. There's no reversal of the what do you say? There is no therapy to the disease. So if you are thinking of a disease with more than 1,700 mutations linked to that, even gene therapy is kind of ruled out. Okay, so in such situations, they came up with a solution to halt the disease progression. So you are having, the mutations that I said, they can be in different parts of the protein. So there could be gating mutations which end up giving you dysfunctional channels which are expressed on cell surface. So channels are expressed, but they are dysfunctional. And there are situations where there is a residual function of the channel. That is, minimum number of channels are expressed on the cell surface, leading to a suboptimal function. Then there is a lack of expression of the protein itself on the cell surface. That is the worst case scenario. So this Vertex Pharmaceuticals was working with potentiators and correctors for the cystic fibrosis. So potentiators were increasing the channel activity, and correctors were trying to increase the CFTR protein expression on the cell surface. So then they did a search with 10,000 molecules, which actually finally gave them what is today sold as Ivacaftor. So Ivacaftor restored the function of this protein CFTR in patients with a specific mutation, that is G551D mutation. So here I would like to touch upon the part of patient stratification. That is the actual part that I work with. Basically, when you are thinking about a target, and even in the early stages of drug discovery, what the pharmaceutical companies these days are doing is to think about a patient stratification strategy. So you are not going to try your drug on the entire CFTR population here. So if what would happen if Ivacaftor was tried on all the CFTR, on all the cystic fibrosis patients? See, this group 1, this is a very small percentage of patients which actually have the mutation. All right. If the company were to go and try the drug on the entire population, you would never get FDA approval for this drug, right? So they tried it on the right set of patients. That is where the patient stratification becomes important. Even from the start of target ideation, people should start thinking of a translational hypothesis or a patient stratification strategy. So yes, all right, you have thought about the target, but even if you get a compound that actually engages with the target, that is, you get the lead discovery happens. Still, what is the set of patients that you eventually want to try it out on is something that is thought of very extensively these days in the early drug discovery. All right. And that is what enables, actually can and enable pharmaceutical industry to get better rates of approval. So now they applied this patient stratification strategy, G551T mutation. The patient should have this mutation, and then the effector is, you know, helpful. So you might want to go back and take a look at how this whole thing works. All right. So how effector functions. It's interesting. So you have actually they started getting promising results enough for pharmaceutical development. An initial set of patients that they tried on showed a very good result where 2 weeks post administration they could see 10 to 12% improvement in lung function. So this what you're seeing here. So untreated patients and ivacaftor treated patients, placebo and ivacaftor, you are seeing the and this is the change in the percentage of post expiratory volume. So the lung function test, all right. So in the lung function test, people started performing better. Okay, so this thus was a hit. So this was a really good drug and has helped a lot of cystic fibrosis patients if they have this mutation. But still, it is at least for those set of patients now we can help, right? So I hope I have explained. So this brings me actually to the end of the webinar, where I have actually shown you how the whole process of early drug discovery happens. Now we just have the lead optimized. Okay, we have a set of maybe 2 or 3 compounds, or a set of compounds which might work, which engages with a specific target. Now the whole story of preclinical development begins. Preclinical development involves testing it out on animals, ADMET, that is the absorption, digestion, excretion, metabolism, and toxicology. So all that happens, pharmaceutical development, including formulation, and all those things happen. So that is another whole set of processes that is yet to happen. So so that is the next step, which actually this webinar doesn't deal with. So this is the part of early drug development that I have covered with an example of ivacaftor. Okay, so from this webinar, I hope you have realized that this is a very complex and multidisciplinary process. So it takes several groups to come together, and this uh, uh, process in recent days has seen extensive application of novel technologies like automation and high throughput screening. They have significantly impacted the process and made the process very efficient. And this is an open area for the application of machine learning techniques. And in the recent years, as I suggested, there is a lot of emphasis on translational hypothesis even from the target identification stage. Also, there is a lot of application of novel technologies and bringing in all these translational aspects, which can actually increase the success rate of candidate molecules. And that is the whole goal of bringing in new approaches into the early drug discovery. Okay, so what I actually, this is what I wanted to share with you. So if you have any questions at this stage, I would like to take them and then go on with the other. Yeah. So somebody asked about vaccines. Yes, actually, I don't have much experience with that. I shouldn't be speaking about what I don't know much, but the process is similar. I can understand that, given the present situation, you would be interested in knowing how a vaccine is developed. Basically, it's hard for me to say that. Let me be honest. I don't want to talk on a topic on which I don't have much experience or insight.
Thank you, madam. We have a very good understanding of whatever you told now regarding the drug targets and all these things.
Okay.
Just addition, I just asked.
No problem. So is there And so basically, but I can tell you one thing: it is the same set of processes that it goes through. So now you can imagine why it is
Yeah.
not that easy to get a COVID vaccine, right?
Okay.
So
Thank you, madam. Thank you.
okay. How can we develop lead molecule if multiple genes or proteins are involved in a disease? Yes, you will have to have multiple. You will have to have a combination approach. So you will have to find what is the combination that works better. For example, cancer is a very ideal example for that. You have multiple signaling pathways which go wrong, and you have things targeting multiple signaling pathways. Or you take a cytotoxic approach where you try and kill the cancer cell, detect the cancer cells early, and try and kill all of them. But still, you have the problem of therapeutic resistance because that one clone which did not get that one cell which probably did not get killed by your uh, one cytotoxic compound still survived, and now it is resistant, right? So basically, that is why combination chemotherapy is the approach. Targeted therapies are also an approach. For example, if there are specific kinases, then you do a combination that you know are implicated. Then you have a inhibitor for that kinase, which is uh, which came up these days, or the inhibitor of angiogenesis plus a kinase plus a cytotoxic drug. So that is how you start combining them. So there was another question which lost, disappeared. What is the most complex and time-taking stage? Target identification, both and lead discovery. Actually, to validate a target, the set of experiments that you do, the whole thing, even recently, what I have heard and what I have gone through, it takes at least 2 to 3 years. And it involves multiple groups. It's not like one PhD student working on it, right? So if you think about it that way, it's multiple groups and multiple people trying to work on that. So, yeah. Actually, the screening and that part, it probably takes a little lesser time. But if the target identification and validation should probably take a bit longer. Yeah.
Do we have any other questions?
Do you have any?
There are no more questions. I would like to propose the vote of thanks to Dr. Jyotsna Rao, and I'm really happy to do that. Thanks, Dr. Charita, for giving the opportunity and introducing Dr. Jyotsna Rao to our students and to our research team. And we are very happy to go through the complete spectrum of how this drug discovery and what are the things that we can do it. And I think the best, the Center of Excellence Computational Genomics from RV is that we can always complement on the bioinformatics side as well as the AI drug discovery, which can make this. So this is very nice of giving her the spectrum, and it was such a nice webinar. Like it was really very so it's giving a lot of insights with a real pace what the students need. So it was not very quick or very slow. The real pace what the students need and what the researchers like us need was completely answered, and it was delivered. Thank you, Dr. Jyotsna Rao. And we are very happy, and there's a lot of takeaways for us and the students. And if you all agree, we can stop the stream and then we can start the hackathon question. If everybody is agreeing on it, we can stop the live stream of in YouTube.
Okay. Thank you, Vidya, and it's very kind of you. I hope I have done justice to the topic.
And
Okay.
we are also looking out for an active collaboration already we have with NS, so we can have a sort of good collaboration where, wherever you want complement from RVCE, our
Yes,
expertise and our thing, and we can always have a collaboration. And this attempt is to have a long-lasting relationship and the problems. If students can solve it, we can definitely have a proof of concept to go about
yeah,
it further. So it can be a start point for everyone. And
yeah, yeah. I look forward to that.
yeah,
Let's see how we can achieve it.
how things, yeah, that's what. Nevertheless, we have started. Let us see how it takes off now.
Yes.
Okay.
Okay.
Thank you.
So shall I? I need to share the hackathon problem. How do I
Yeah,
do?
we will. One second, we'll stop the YouTube stream.